we have lesson 3, and the name of the lesson we will study today, studying the human genome. So today we are going to take what? Today we are going to take the human genome. Okay, so if you look to the topic, right? So it is about the internal study of gene, right, or genome. Mean the entire genome how we will take. So if you look to the previous chapters, the previous lesson was human chromosome. First, we have studied human chromosome. Second, we have studied human genetic disorder. And the last lesson of this chapter we have, it is studying the human genome, right? Here in this lesson, we will study a human genome. Just you have to think about some facts which you have here. Just a few decades ago, right, computer were genetic machine found only in laboratories in universities. So before decades, right, a few decades, computer was only a genetic machine that was found only where in the laboratories and university. But if you look to today life, today many of us carry small, powerful computers to school and work every day. So if you look to your mobile phone, so it is also an example of computer, right? Decades ago, the human genome was unknown. So if you think back, let's suppose a few decades before, the human genome was not known. It was unknown. Today we can see our entire genome on the internet. But today you can see the human entire genome on the internet. How long will it be? How long it will be before having a copy of your own genome is an ordinary as carrying a cell phone in your pocket, right? So now it is not difficult here, right, to check your genome, right? And you can even what are the entire genomes of the human, right? First point in which which we will study in human genome is manipulating the DNA, right? Manipulating the DNA. So what techniques are used to study a human genome? First thing, if you are going to study the human genome, so what are the techniques with the which way we are going to study a human genome? Technique we will use, right? What will be the technique? With the help of that technique, we will be able to study the human genome. Okay, by using tool, right? By using tool that will cut, separate, and then replicate DNA bases, right? Base by base. Scientists can now read the base sequence in the DNA from any cell. So now the scientists reach to a level that they can do what? They can study the entire genome. But what are the techniques we are using for studying the genome, right? What are the techniques? So are the techniques will include what? The techniques will include tools and techniques, tools and procedures, with the help of which we will do what? We will be able to study the DNA. So what will be the tools and what will be the procedure? So tool, we have different machines now, right? As we are using computer, we are using ELISA. Different. So in this case, we are using what? We are using different machine now. Is to cut and separate and then replicate DNA bases, right? Base by base, mean one base, another base, and another base. Scientists can now read the base sequence in DNA from any cell. If you take one cell of your body, right? So you can take the entire genome of your body from one cell, right? This is possible due to what? This is possible due to the the human genome, the studying human genome. Okay. Now, DNA is a huge molecule, as it's a macromolecule. We are saying macromolecule mean a large molecule, right? Even the smallest human chromosome contain nearly 50 billion base pairs. So, if you look a small human chromosome, if you take, right, if you take the smallest human chromosome, so it will contain round about 50 million base pairs, right? Manipulating such a large molecule is extremely difficult. If you are going to manipulate, if you are going to explore, if you are going to study such a molecule, right, which is a roundabout 50 million base pairs. So it will not be easy, right? It will be quite difficult. In in the 1970s, scientists found that they could use natural enzyme in DNA analysis, right? So in 1970s, scientists found that they can use natural enzyme DNA analysis when they want to explore the DNA. So they can use what? They can use the natural enzyme. Scientists can now read the base sequence in DNA by using these enzymes, right? So now the natural enzyme are used by the scientists. For which purposes? For the purposes of what? For the purpose of to cut the DNA, to separate the DNA, the different segment of the DNA, and to replicate DNA bases, right? And to replicate DNA base by base, right? Fraction by fraction. Guys, can you see the screen full?
We can.
Okay. I don't know. I have logged in with my mobile phone, so when I'm changing side here, so it's not going to change. So that's why I asked. Okay. So first thing which we will do in the DNA, right? How we are going to
Mister,
manipulate the DNA?
Sir, I'm
Yes,
seeing the
please.
screen in blue only.
we only see one photo on the screen share.
Okay, Kazim, you also have the same problem.
What problem?
Which slide do you see on your screen?
The first one, biology lesson overview studying the human genome.
So I have already finished in manipulating. What is the problem? Wait me just 1 minute.
Well, we can read the books.
Just let me, because I have to change from here. So you must need to. You can see now cutting the DNA, right?
Yes, cutting the DNA. Cutting DNA.
Still, it's not changeable. Okay, so it means that I have to not to maximize it. I have to go with this screen. When I maximize, it's not working with you guys. Okay, so if you look right to the what are the techniques? So the techniques, the manipulating of DNA, we will start from what? We will start from the cutting the DNA. So how we are cutting the DNA? What are the biological scissors of the for what will be the biological scissors we will use for cutting the DNA? So nucleic acid are chemical chemically different from other macromolecules such as protein and carbohydrates. So if you look to nucleic acid, so they are not same as like what? They are not same as like the other macromolecules such as protein and carbohydrate. This reference makes DNA relatively easy to extract from cell and tissue. So if you look to the cell and tissue, right? So and it is different. Nucleic acid means DNA is different from that of the protein and from that of the carbohydrate. So it is different. Then it is very easy for us that we can separate DNA from the rest of the mean from the rest of the carbohydrate, from the rest of the protein. DNA molecules from most organisms are much too large to be analyzed. So if you look to a DNA molecule, right, from the most of the organism, it is what? It is too large that we can analyze. As I told you that it will be round about how much? It will be round about 50 million base pairs if you take even a single smallest chromosome. So they must first be cut into smaller pieces. So if it is very large, right? If it is very huge amount, so we have to cut that huge amount into small fractions, right? Into small what? Into small fraction. How we will cut it into small fraction? Many bacteria produce restriction enzyme that cut DNA molecules into pieces called restriction fragment that are several hundred base in length. So we have a bacteria and that produce an enzyme, and that enzyme is called restriction enzyme. What is the function of that restriction enzyme? The function of the restriction enzyme is that it can cut the DNA into the DNA fragments, right? This process we call restriction fragments. And that fragment will be of how much length? As we have the entire up to, let's suppose, 50 billion or more, sorry, million or more. So then that fraction will be of several hundred bases in the length. So mean it is now reduced, right? It will not be as lengthy like in billions or like in millions. So now it will be short. So if you have a chart, so then you can study it easily. Okay. One of the hundreds of known restriction enzymes, such as each cut DNA at a different sequence of nucleotides. So we have a lot of restriction enzyme, right? So restriction enzyme, specific restriction enzyme will cut DNA into a specific sequence, or we can say into different sequence of nucleotides, right? As we have different type of restriction enzyme. Okay. For example, if you remember the example we have studied E. coli, right? E. coli we have studied in the previous chapter. So, for example, the EcoRI restriction enzyme recognizes the base sequence GATTC, right? So, we have this eco R1 or right? These are the restriction enzyme which will recognize the base pair of only what? Which base pair composed of guanine, adenine, thymine, and cytosine, right? In this sequence, so the eco R1, it will recognize what? It will recognize only this sequence. It cut each strand between the guanine and adenine bases. So where it will cut the bases into guanine and adenine bases, leaving single strand overhang called sticky end. So it will, what will be the remaining? The remaining things that will be a sticky end, and that will have a sequence of AATT, right? So this will be the sequence which will be remain. And this sequence, when you see this sequence, so it means that it will be, but it will be a sticky end. We call it sticky end. The sticky end can bond or stick to DNA fragment with the complementary base sequence. And to this sticky ends, it can bind to the DNA fragments with the complementary base sequence, as you can also see in the diagram, right? Here you can see, right? Addition of restriction enzyme EcoR1, right? So this is the DNA strand where it will attach, right? It will attach, as we said, that at GAATTCC, right? So GAATTCC. So here it will bind, right? At this point, it will bind. I mean, it will break the DNA into fragment. And after, what will create? It will create a sticky end. This is called sticky end. This place will sticky end, which will happen, to which the eco R1 can bind again. There. So this is what? This is about the cutting the DNA. In which way the DNA is going to cut? In this way, right? That we have restriction enzyme. It will cut what? It will cut the DNA into what? It will cut the DNA into its fragment. Let me check from the book. Cutting the DNA. Okay, you can see in the book on page 404, right? On 404, the diagram you can see on 404, that is quite that diagram is quite good, right? If I have here, so I will share with you, just open from the book, then I can share with you. Restriction, you can see right from the book on page 404, figure 10.1011. The first one you can see cutting DNA, right? So a restriction enzyme is like a key that fit only one lock. Restriction enzyme is what? It will act like a key, which will fit only one lock. The eco R1 or RI restriction enzyme can only recognize the base sequence of, as I told you, guanine, adenine, thymine, and cytosine. It will cut each strand of DNA between the guanine and adenine bases. and the living will be the living end. The remaining end will be what? The remaining end will be it will we called it sticky end, right? And what is the function of sticky end, right? Because they can bind or stick to DNA fragment with a complementary base sequence, as you can see in the book also, and the diagram which I show you from both of it you can see, right? So this is clear till now, right? Till this place it's clear. Clear, boys?
Clear.
Any else student else, if you have question, you can ask me now. Then we are moving to separate DNA. Okay, so we will move to the DNA sequence. Sorry, not the DNA sequence. Reading, separating the DNA. How we are going to separate the DNA, right? I will share also with you the video of this, right? So you can go through it also. Separating the DNA, right? Now that was just cutting the DNA. I mean that how you are going to cut the DNA into fragments, right? But as far as I think, the important is what is the important? The important here we have it is what? It is how we are going to separate the DNA. Like if you take a part of the human, let's suppose a human or anything, right? Like blood you have taken, like a tissue you have taken. So how you have to extract the DNA from this? So this is important, right? Separating the DNA, or in other words, you can say DNA extraction. How you are going to separate the DNA? How you are going to extract the DNA in the pure form? So this is a very interesting process, right? But here you have to be attentive to this point. One DNA has been cut by restriction enzyme. Scientists can use a technique known as gel electrophoresis, right? So the technique by which we can extract or we can separate DNA are called what? Are called gel, right? So gel, it we are using that is a medium we are using for it, right? That medium we call gel electrophoresis, as you know that it is process, right? So gel electrophoresis to separate and analyze the different size fragment. You can see on the diagram, right? But you will not understand now. Uh, Before this, also some process to which we have to purify DNA. Then we will come to electrophoresis, right? So how you are going to separate the DNA, right? Going to separate the DNA fragment or a DNA. A mixture of DNA fragment is placed at one end of the porous gel. But this is after when you extract the DNA, right? When an electric voltage is applied to the gel, DNA molecules which are negatively charged move toward the positive end of the gel. When you put the DNA into the porous gel, right? And you applied after that electric current, right? Or electric voltage to their gel. So what will happen? The DNA molecule which are negatively charged, as we know the DNA molecule are charged, so it will move toward the positive end of the gel, right? So mean if it is negative, they will go to the positive. From chemistry point of view, we know this, right? The smaller the DNA fragments, the faster and it moves. So if the DNA fragment are small, they will move faster and they will move further. Separating, so separating the DNA, the result will be what? The result is a pattern of bands based on the fragment size. Specific strain that bind to DNA make these bands. So there will be a specific strain, like specific colors will be here, or specific strain with there, which will bind with the DNA and it will make these DNA. We have studied marker, right? Markers we have studied. So that markers we
Genetic
are
marker. Yes, we did.
genetic marker. So the genetic marker are some other marker which will use in the DNA. So that marker we are using, and with the help of that marker, we can see the bands of DNA visible, right? Researcher can remove individual restriction fragment from the gel and study them further. So this is if you want to take its picture, so you can also take picture, and the picture will be as same like if you see in the Watson and Crick model, right? Now, what will be the reading of DNA, right? So the reading of DNA is based on the DNA sequence, right? Sorry, the nitrogenous basis. So if you remember in the previous chapter we have studied, so after the DNA fragment have been separated, researcher can read the sequence, right? So now if you have separated and it's visible, right? You can just read its sequence. Single strand DNA is placed in a test tube containing DNA polymerase. What are DNA polymerase? DNA polymerase are enzyme which perform different functions. Like for example, unzipping of DNA, breaking of DNA, right? And even it help in the replication of DNA. It help in the replication with the DNA. The enzyme that copies DNA along with the 4 nucleotide base adenine, thymine, guanine, and cytosine, right? This will occur in the DNA. The function of polymerase enzyme here we have to copy the DNA, right? It will copy the DNA. The DNA polymerase uses the known strand as a template to make a new DNA strand after another. It will be used for what? The DNA polymerase will use the unknown strand as a template to make a new strand of the DNA. Okay, researcher also add a small number of bases that have a chemical dye attached each time a dye labeled basis is added to a new DNA strand. The synthesis of that strand stop. So researchers are also going to add a small number of bases, right? That we have a chemical dye, right? So it will be used for what? It will be used to label the bases, right? Which will be added to a new DNA strand. When the DNA synthesis is completed, the replication of DNA completed, the result is a series of color-coded DNA fragments of different length. So you will get when the replication is completed or the synthesis is completed, so you will get a colored coded DNA fragment, and the color coded DNA fragment will be due to what? Due to the dye and due to the marker we have used in it, right? Researchers then separate these fragments often by gel electrophoresis, right? So these fragments can be separated after by what? After by gel electrophoresis. The order of the color bands on the gel tells the exact sequence of the base in the DNA. So the color, as I told you, the color will tell you the colors, or the marker will tell you what? It will tell you the exact sequence of the bases of the DNA, which you can see also in the diagram. The entire process can be automated and controlled by computer, so the DNA sequence machine can read thousands of bases in a matter of seconds. So as I told you that we are using different machines like computer, we are using DNA electrophoresis gel we are using, and beside this we are using PCR machine, right? Polymerase chain reaction machine, which we have in the next point, right? So if you have any question till this place, you can ask me. And this lesson is almost finished. The next lesson we will start from the Human Genome Project, I think. Yes, we have Human Genome Project. If you have any question, you can ask me here. If you have any
No
questions.
questions.
No question.
Okay. Okay, if you don't have questions, if you want, you can leave.